Hello. Right now we're going to go over chapters 6.1 and 6.2. We're talking about the tools of biology and cell size. Hopefully this is a review for most of you. And in 6.1 we talk about the tools of a biologist, or the tools that we use in order to study life. And the first of those is a microscope. And so let's talk a bit about a few of the different microscopes that we use in class, or not necessarily in class, that we use in biology. The first microscope that we're going to talk about is a light microscope. And so that uses light waves. So if we look at this picture here, there's going to be light that goes up, and the light's going to bounce off the specimen, and it's going to go through these convex lenses, which is going to make it bigger. So these lenses are like magnifying glasses here. So the light is going to bounce off of our specimen, go up into the lens once, get expanded, and if it's a compound light microscope, compound meaning double, then you're going to have another lens here. It's going to expand it again. And so these light microscopes tend to have a ratio. It's going to give a magnification of about 1,000 times, which means that if you are looking at something that's 1 centimeter large in life, then it's going to appear about 1,000 centimeters. But there are limits to this tool, that it can only show specimens that are that are larger than a light wave. So if you get smaller than 400 to 700 nanometers, the light isn't going to be able to bounce off and go into your eyes the way that it would if it was larger. So the limiting factor of a light microscope is the size of the light waves. And so we talked about magnification. So that light microscope, compound light microscope, usually has a magnification of about 1,000 times. And so resolution is another word that we're going to use when we talk about microscopes. And that's basically just the clarity of the image. Do 2 points have to be in order to, or how close do they have to be in order to realize that they are distinguishable between 2 different points? I like to think of this little game when you have 2 sort of sharpish objects. When you put them close together on the back of your hand, you can't really feel that it's 2 separate objects until you pull them apart a little bit, and then you can feel that it's 2 different objects. And that's sort of the same concept as resolution. How far apart do the points have to be in order to distinguish that they are 2 separate points? And finally, our last vocab word is contrast, and we have it's the difference between the light and the dark. So can you tell the difference between the light and the dark? And oftentimes we have to increase contrast by using dyes. So here we have dyed the DNA in this cell as it's going through mitosis, so that we can see it more clearly, or with more contrast from the rest of the cell. The next microscope that we're going to talk about is electron microscopes. And there's 2 different types of these microscopes. And these microscopes are going to help us to see the very small things, the things that are smaller than the waves of than the length of light waves. So these were developed in the 1950s, and what it does is it uses a beam of electrons aimed at the specimen, and it can show us our organelles. So it's going to bring us into the range of 0.002 nanometers to about 2 nanometers across. And so this is much smaller than our compound light microscope. And so there's 2 different types, as we have said: the scanning electron microscope, also known as the SEM. And this shows a 3D image of our specimen. What we do is we put a gold plating on our specimen. So this is an image that was created by a scanning electron microscope, but then color was added afterwards. So we don't develop the color with the microscope. That is added after. But so we put gold plating around on the specimen, and then we scan or spray a beam of electrons, and it's going to create a 3D image. So those electrons bouncing off of the specimen is going to create the image that we see here. And the transmission electron microscope is different in that it's going to show us what's on the inside. So the scanning electron microscope shows us the shape whereas the transmission electron microscope is going to show us what's on the inside. And what we do is we coat internal structures, perhaps organelles, with a coat of heavy metals, and the electrons are the beam of electrons are going to spray down onto the specimen, and it's going to not be able to go through the certain areas that are coated with heavy metals, and so it's going to show us a shape of the structures that are on the inside of our cell. And so these are our two electron microscopes, SEMs and TEMs. Another tool that we use during the study of life is our centrifuge, and we have a centrifuge right here. And the centrifuge is a tool that spins the sample at high speed. So I will put, I will put test tubes or the samples into the centrifuge, and then it's going to spin at very, very quick rates. And what happens is the centripetal force is going to pull the heavier materials to the end of the test tube. And so we can see here after 10 minutes, this is a test tube that's been in a centrifuge for 10 minutes. The heavier materials like the nuclei and the cellular debris are going to form something that we call a pellet at the bottom. And so the rest of the material is called the supernatant. And so if we're trying to separate materials in a cell, we're going to have to go about separate density. And so when we want to get the less dense materials, we're going to pour that supernatant into another test tube, run it again. So you can see after another 10 minutes or 20 minutes total, you're going to have mitochondria and chloroplasts. And if we take that pellet out and dump that supernatant into another test tube, we're going to have less dense materials, smaller and less dense materials. So that is our centrifuge. As we're studying life, we have to remember, and we're using these tools to study life, we have to remember that life is broken up into three distinct domains. Prokarya, also known as bacteria, archaea, which is another single-celled form of life, and eukarya, or eukaryota. And this is the tree of life. On eukaryota, we have more complex organisms, multicellular organisms. We have kingdom Animalia, kingdom Plantae, kingdom Fungi, and kingdom Protista. And while it's important to know that there are many other kingdoms, these are the ones that we're really studying and understanding that exist in eukarya. And so we talk about how eukaryotes are multicellular. They become a bit more complex, or a lot more complex, than prokaryotes. And really, one important characteristic of complex organisms is the compartments in which you have within the cell. And so if we look in this cell here, this is a eukaryotic cell, and you can see that there are all of these different places. And here we have the nucleus, and then we have the cytosol, which is the space existing in the cell, and then you have the inside of the Golgi apparatus out here, and the endoplasmic reticulum, and then you have the inside and the outside of the mitochondria. All of these places have different pHs, different salinities, perhaps different temperatures. And so this creates concentration gradients and a mechanism in which to move materials from one place to another and to allow for chemical reactions to occur. So these compartments are very important in allowing life to become more complex. A eukaryotic cell, you can see that they have non-membrane regions surrounding the protein. So there is no nucleus. There is a nucleoid region where the DNA exists, but there isn't a compartment for it. Respiration does occur, but it occurs in folds in the plasma membrane, not within an organelle. So there aren't any membrane-bound organelles within a prokaryotic cell, but there are but they do exist in eukaryotic cells. And so here it's these plasma membranes, these lipid bilayers, that create these barriers that allow for a different pH to occur, or a different salinity in one place or another. And then within these plasma membranes we have proteins that allow for ions and molecules to travel through, and allows for these chemical reactions to occur, and so complex life can occur. The last concept that we're going to cover that helps life to become more complex is the amount of surface area that organelles or organs are going to be able to use. And so the more surface area that an organ or an organelle has, the more opportunity it has to interact with chemicals or allow for chemical reactions to happen. If you wanted this cube to get wet, saturated inside and out, you could dump water on this cube, or you could dump water on this cube, which has increased surface area exposed to the world. And so if you were to dump water on this cube, it's going to be able to saturate it completely a lot more quickly than it will this one. And so that concept is used again and again and again in life. So if we have our trachea here, and we have bronchi here, the bronchi are going to have bronchioles extending off of it. And there's going to be extensions off of the bronchioles, which are going to increase the surface area even more, which is going to allow for the amount of oxygen or carbon dioxide that's interacting with these lungs to, the amount is going to increase significantly, as opposed to if you just had two bronchi and no extensions off of them. So the surface area of a cell or of an organ is very important, and it's a concept that life has taken advantage of over and over again. I hope this helps, and have a great day.